Right now, as you're watching this, inside trillions of your cells, there's this microscopic engine just spinning away. It's called the Krebs cycle. You might also hear it called the citric acid cycle. And honestly, it's one of the most fundamental processes for life as we know it. So let's dive into this amazing metabolic machine and see how it works.
It all really starts with a simple but kind of profound question, right? How do you turn the food you eat, say a peanut butter sandwich, into actual usable energy? How does that lunch you had fuel your ability to lift a weight or run a mile or even just think? The answer is this beautiful multi-step process happening on a tiny cellular level.
Well, the big picture answer is something called cellular respiration. That's the catch-all term for how your cells take the energy locked in food and harvest it. And the whole point of this entire operation, the grand prize, is to create molecules of ATP. You should absolutely think of ATP as the energy currency of your cells. It's the cash they need to pay for pretty much everything they do.
But here's the thing. Turning a sugar molecule into a big pile of ATP isn't just a single magic step. It's much more like a highly efficient four-stage factory assembly line. Our focus today is on stage 3, the Krebs cycle, which is really the central engine, the powerhouse at the heart of this entire process.
So here's the roadmap for our factory. First, you've got glycolysis, which breaks down glucose. Then there's a quick connecting step called pyruvate oxidation. And right there, smack in the middle, is our main event, the Krebs cycle. It does a lot of the heavy lifting to set up the massive energy payoff in the final stage, which is called oxidative phosphorylation.
Okay, so how does the fuel from our food actually get into this cycle? It can't just barge in, right? It needs a special kind of entry ticket to get past the gate. And that ticket, that golden ticket, is a molecule called acetyl-CoA. Remember that pyruvate from stage 1? Well, it gets trimmed down from 3 carbons to a 2-carbon group, which then gets attached to a carrier. And this new molecule, acetyl-CoA, is the crucial link. It's the one that officially gets the party started. So now we're at the main event, the heart of it all. We're going to take a little tour of the cycle itself. And hey, don't worry about memorizing every single molecule name along the way. That's not the point. We're going to focus on the big ideas, the beautiful logic behind this whole process.
I know what you're thinking when you see a diagram like this. It looks complicated, and you're not wrong. But trust me, the whole thing really just boils down to a beautiful and logical shuffle of carbon atoms all perfectly coordinated by 8 different enzymes deep inside your mitochondria. Let's break it down into 4 key moves.
The first move is simple. Entry. Our 2-carbon acetyl-CoA comes in and hands off its acetyl group to a 4-carbon molecule that's already waiting in the cycle. This molecule is called oxaloacetate. They link up and bam, you get a new 6-carbon molecule called citrate, and that's actually why this is also called the citric acid cycle.
Next up, release. In a couple of precise steps, 2 of those 6 carbons get snipped off and released as carbon dioxide. And get this is the really cool part. The CO2 produced right here is literally the exact same carbon dioxide that you are breathing out right this second. It's the exhaust from your cellular engines. So when you break those carbon bonds, a bunch of energy gets released. But it doesn't just float away as heat. No way. It's captured by these special molecules, NADH and FADH2. Think of them as little rechargeable batteries, or maybe energy taxis. Their whole job is to pick up these high-energy electrons and shuttle them over to the final stage where they'll be used to make a ton of ATP. And finally, the last move, regenerate. After we've kicked out 2 carbons, we're left with a 4-carbon molecule. The last few steps of the cycle are all about shuffling this molecule around to get it back to its original form, that oxaloacetate we started with. This is what makes it a true cycle. The starting line is restored, ready and waiting to grab the next acetyl-CoA that comes along.
All right, so we've done a full lap around the track. Now let's stop and actually count our winnings. What's the actual payday from this central engine for every 1 glucose molecule that we started with? Now, first thing to remember, 1 glucose molecule gives you 2 pyruvates, which means you get 2 turns of the cycle. From those 2 turns, we get 4 molecules of CO2 as waste, and only 2 molecules of ATP directly. You might be thinking, that's it? But hold on. The real prize here, the big win, are those 8 high-energy carrier molecules, the 6 NADH and 2 FADH2. These are the guys that are going to go on to make the real energy fortune in the final stage. But the Krebs cycle is so much more than just a dumb wheel spinning mindlessly to break down fuel. No way. It's an incredibly intelligent and responsive system. It's basically a metabolic master switch.
You see, it has this brilliant feedback loop built right in. When your cell is running low on energy, which means it has lots of ADP lying around, the cycle speeds up. But when the cell is flush with energy, with plenty of ATP and NADH, key enzymes get shut down, and the whole cycle slows right down. It's just like a smart thermostat, perfectly matching energy production to what the cell actually needs in real time.
And this is where it gets even more fascinating. The Krebs cycle isn't just about demolition, breaking molecules down, a process called catabolism. It's also a construction yard, a hub for building molecules up, which we call anabolism. And because it's a two-way street, it plays both offense and defense, so it gets this cool name, an amphibolic pathway.
So what does that mean? It means that molecules can be pulled out of the cycle at different points to become the raw materials for some of the most essential building blocks of life. We're talking about amino acids for proteins, fatty acids for your cell membranes, even the parts needed to build your DNA, RNA, and the heme in your blood. It really is the absolute central hub of all metabolism. So, we'll end on this final thought. This elegant, efficient, dual-purpose cycle. It's not just in you. It's in a mouse, a fish, a tree, even in yeast. Its nearly universal presence across almost all life that breathes oxygen tells us it must have been one of evolution's earliest and most successful inventions for managing the energy of life. So what does that say about our shared biochemical heritage? Pretty amazing stuff.